To get a soil core, which is the first step in ascertaining what is the mass of carbon in these sites, one first puts a core into the ground, extracts the top meter of soil. That's a specialized soil sampler for mangrove ecosystems, which is beautiful at extracting a pretty large volume of soil that's relatively undisturbed. At which point in time, once you extract it out of the ground, we separate the soils by depth because the carbon concentration may vary by depth. And so what we're doing here is what is done really throughout the world, is sampling the top 15 centimeters, 15 to 30, then another sample at 30 to 50, and a sample in the midpoint between 50 and 100 centimeters. So we would take those, knowing the volume, to the lab to do the laboratory analysis. And then you might want to sample the next meter down, meter 1 to 2, and even meter 2 to 3, up until bedrock, till you would hit a indurated or a historic surface, and that would be the total carbon stock. There is going to be some variability as you move throughout the forest stand here. And so what we would recommend is 5 to 6 samples per site. That way you have an idea of the variation and the standard error of the sites. But we have found that 5 or 6 cores is sufficient to get a very accurate estimate of the soil carbon stock in a mangrove ecosystem.